Alright, so this is for H8.2, cells. I think it's like cell structure. You know, it's like cells in microscopy. It is 6 p.m., so I'm going to try my best to make this quick. And so, to replace my ugly ones that are on Canvas, first let's do our little diagram. This is page 18. And so this is for prokaryotic cells and prokaryotic cell structures. I know that you all did your coloring sheet and that you've had access to the little, little click and learn. But I know sometimes it's hard to kind of visualize these things, so I wanted to be able to do a little practice sample drawing for ya. So when you have a prokaryotic cell, you have some things that are always there, and this is true, I guess, with any of your cells. You have some things that are always there for surezies, like you can't have that type of cell without them, and then you have some things that are like sometimes there. And so mine has like 3 beautiful layers, pre-drawn, so that you don't have to sit here and watch me struggle. But they're typically pill-shaped, at least with the bacillus type. They have like different types of bacteria and different types of prokaryotic cells that'll come in different cell types. But your most common one that you'll see is your bacillus bacteria. So here she is in all of her glory. And so the first thing is the outermost layer that some of your... There we go. That some of your bacteria will have is a capsule. So not all of your bacteria are going to have a capsule, but some of them will. Just kind of like an outer additional coating. This is your capsule. For things that are sometimes there, I'm going to put a little star next to them. Your like next inside layer. We'll make it... I'm going to run out of colors real fast. I'm going to be annoyed. But we'll make our next layer our cell wall, which we'll do in orange. You know, I drew the little lines to trace, and then I'm not tracing them. Good job. Why did I... Oh, I should not have picked orange, because I don't have an orange highlighter. It's okay. We'll just color it in real quick. Da-na-na-na-na-na. Oh, jeez Louise. But that is your cell wall. It protects the cell and helps maintain the shape. Cell wall is always there. Your capsule is not necessarily always there. So your capsule is like a double layer for some of them, but if it's not there, your cell wall does all the protecting, all the maintaining of shape. When your capsule's there, very similar vibe, very similar role. The next layer in, let me pick a color I have a highlighter for. We'll make it I guess we're going to make it green. I said purple, and I picked up green. Is your cell membrane. This is not drawn to scale. The cell membrane is not this thick. It also is not ugly like that. Why did it... Okay. Well, now I'm realizing this was a dumb idea, because when I color it in with a highlighter, it's going to look goofy. Oh well. It's fine. It's okay. I'm not restarting this. It's okay. There we go. Beautiful. That's your oh, where am I going to put this label? It's your cell membrane. Someone's calling me. Do I answer it? No. And it allows entrance and entrance. That's wrong. And exit of substances. Spell entrance right on yours. And then the other thing that kind of is on like the outside of your cell is you have these like pili, which are technically just protrusions of the cell membrane, but I'm gonna make them a different color just so that you can tell the difference. But they're really just like outgrowths of your cell membrane. They kind of make it look hairy. They're gonna make this drawing a real beaut. Oh, how gorgeous. And so these are your pili. They're only sometimes there. They're like hair, leg extensions. Extensions. That's wrong too. Great job, Jessica. Used for attachment of cell to substances. They're like little grabby hairs. Like one of those like sticky balls that like you throw at the wall. That's them. Oh, I did have a description for capsule. I just didn't notice it because I wrote so ugly on the first one. Oh, I described it in a really gross way. It's slimy layer. Protects cell and adds to virulence. Oh, that's why. I remember why it was important. If it has a capsule, it's more likely to be able to spread throughout your body. Like, it can help it be more, like, resistant to antibiotics and stuff. So, that's why I wrote that. I remember now. Now I'm cooking. Okay. Let's see, what other colors do we have? The flagella is not that important. It can be pink. No, I got purple. Why? What's going on? Okay. So your flagella, again, not always there, but when it is, it's used for movement. Similar to like sperm. That's hard to tell that it's different than the red. Do I have a pink one? I don't think I have a pink highlighter. I think this is as close as we're gonna get. I forgot it looked like that. Oh well. It's too late now. Alright, what else? We'll do plasmids next. Yes? Yes. So your plasmids, I'm just gonna make mine look like that. It's like circular strands of DNA carrying additional genes. My example is like antibiotic resistance. Oof. Because we made our plasmid blue, we'll make the nucleoid blue too. Meow. So ugly. I'm so sorry. So that's our nucleoid. Again, present in all of them. It's just like the space where genetic material in the form of like naked DNA or like unwound, unprotein bound DNA. No nuclear membrane. It's very loosey-goose in there. Color it in real quick. Beautiful angel. And then, ew, okay. It's fine. And then last up, your ribosomes. I'm going to really regret these color choices because I was going to try to keep them consistent. Okay, we'll write it down here. So these are your 70S ribosomes. They're just protein-producing structures. And they're 70S, they're smaller than in eukaryotes. So we're just gonna write small. I have space. And then for cytoplasm, it's just gonna be yellow. So I'm just gonna color it in. You know what? We're gonna write in yellow too. I don't care. Being a rebel today. Brr, brr, brr. Cytoplasm. It's your jelly liquid. It contains enzymes for reactions in cell. And ta-da! There you go. There's your beautiful prokaryote. I don't know if this is really better than the other one or worse, or the same. They're kind of equally as horrid. I'm sorry. Alrighty. You have this page to do. I'm going to leave you all to do most of this as practice on your own. I feel like the only one that's really, like, hard to figure out is why your Clostridium appears, like, the cytoplasm of your Clostridium appears dark in your electron microscope scans. Forgive me while I flip weirdly through my packet. And so, bum-ba-dum. For your labels, I was lazy, and I feel like the easiest way to label them is just to write the number next to the thing up here. Oh, I don't remember what colors I used for it. Oh, geez Louise. What color was... The cell wall was red. I do remember that one. No, that was the capsule. What color was the cell wall? Orange, because I had to color it. So your cell wall is shown by number 2. Wah! Your cell membrane in this one, which I think was green, is shown by, like, number 1. Your cytoplasm, arguably you could argue maybe it's pointing to a ribosome, but it's too small to really be able to tell. You could argue it's pointing to a ribosome, but, like, it's like asterisk. Maybe, if you really want to be, like, petty and annoying. And then your nucleoid, it's in the middle. It's that big, lumpy fella. And so what causes your cytoplasm to appear so dark in the electron micrograph? So when they stain it, staining, the heavy metal, like, stain that, oh my god, the heavy metal stain they use stains proteins a lot darker, and because this is a bacteria that's used to make a neurotoxin, it's protein based, it has a lot of it stored in its cytoplasm, so it appears darker. Ta-da! I think the other thing would be, like, number 3 is what it would look like if it was cut the other way. So you're looking at it cut, like, sideways. And so what would it look like if you cut it the other way? Imagine it like a pill or like a hot dog bun. So it's kind of like this. And like the way like the way that we're looking at it. And so the way it's cut in this picture is like if you took your hot dog bun and you had like, here's the bottom bun, and then you sliced it like this. See? Hot dog bun style. So if you cut it the other way, you cut your hot dog bun like this, it would just be a circle. Because that's just how 3D shapes work. It's like if you cut a tube, here's the like butt end, it would just be a circle. I don't think I explained that very well. But like, it's just like 3D shapes. I don't know. If you don't get it, ask me in person. I'll cut something for you. These two do on your own. I'm not doing math for you. Sorry. Actually, I'm not. I don't feel like doing math right now. I'm tired. All right. Time for the fun. More fun drawing pictures together. I know you're so excited. Okay. So a lot of these have similar structures, so we can kind of whip through some of the ones that they have in common at the same time. And so I'm going to weirdly start with the nucleus. Don't ask. Just the way my brain is feeling today. And so all of them have a nucleus. Even the little one. So everything highlighted in blue, even the additional things I'm going to add in, they're all nucleus. All of it is part of nucleus. Anything in the blue highlight, nucleus. Just have to label this one too. Okay. I'm not labeling the details on this one. I'm letting you know that right now. That bad boy is way too small. I'm gonna run out of space. Alright. And then within your nucleus, you have several different pieces. You have your nucleolus. Boop. Nucleolus. We'll draw it down here on this one for no reason. Nucleolus. Nucleolus. I'm not labeling it, but there she is. Oh, geez, I need, no, it's fine. You also have your DNA. Okay, there she is. So your DNA. In the form of chromatin fibers. And before, like cell division, they form chromosomes. Which hopefully you're somewhat familiar with. And so, chromatin. We have to draw in your nuclear membrane. So it's like a little, I'm going to draw it in as like a dotty line. This little one's gonna bother me. And so where the line is your nuclear membrane. Fun fact, it's just a membrane that surrounds your nucleus. Nuclear membrane. And then like the gap in between them, those are your nuclear pores. Nuclear pore. They like let stuff in to your nucleus. So nuclear membrane. Oh, there's a gigantic nuclear pore over here because Miss White can't space out a dotted line. Nuclear pore. Nuclear membrane. We'll squeeze it in. Nuclear pore. Okay. Beautiful. I know you can totally tell what's going on. Okay. What's next? We'll do cell membrane next. What color was cell membrane on the last one? Green. Oh, that's unfortunate. It's fine. Alright, so cell membrane. Here she is. On animal cells surrounding them, you only have cell membrane. There we go. Careful, 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 careful. This one looks like a snowman. Okay. We'll put it down here, I don't care. Cell membrane. We also have a septum, which is like a partial division. Oh, we didn't even talk about this top piece of this guy yet. It's okay, we'll get there. He can come later. I'm putting him to the side for now. Alright. I think that that's all the ones that are the same? No, it's not, I lied. The cell wall. I'm not coloring in the cell wall. You can't make me. Here's the cell wall. It's just gonna get too chaotic if I try to color this in. So this is cell wall. In plant cells, it's made of cellulose. In fungal cells, where am I? I'm gonna put it up here. It's made of chitin. Beautiful. And then we have our rough and smooth ER, which is one of my favorites. Do 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 do. And put the smooth one over here. I guess you can have an ER too. I'm not labeling it though. And so, it's hard to tell. Oh, I didn't give you a smooth ER. Sorry. Sorry, fungal cell. It's hard to tell right now. But what makes them different is the inclusion of ribosomes. Your rough ER has membrane-bound ribosomes. Your smooth ER does not. Don't give it any. But your rough ER, it gets to have them. So I'm going to write the label with a pen. Where's the light purple pen? I threw it over here. So this is your rough endoplasmic reticulum, from now on known as the RER. This is your smooth endoplasmic reticulum, from now on known as the SER. Here's your RER. Here's your SCR. your RER, oof, and your SER. Smooth ER does like lipid synthesis, rough ER protein synthesis, hence the ribosomes. Alrighty, what else? I guess we'll do the Golgi. So they're all kind of like lumpy little membraney things. And so here's your Golgi. And it's forming little vesicles for transport. Sending things in and out of the cell. Look at it go. Dun, 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 dun. Oh, we need lysosomes. Okay. Lysosomes will be ugly doo-doo dark brown. Oh, that's gonna be too similar to the ribosomes. Oh no. Oh well, it's too late now. Alright, these are lysosomes. They're very different from the ribosomes because they're big. Lysosome. These do like waste removal. Oh, look at this little maneuver I'm about to do. Bam. Lazy. Okay. We have mitochondria. They're gonna be gray. That's one of the few colors I have left. One of the easy ways, if you're like looking at a picture, to tell a mitochondria is that it'll have little like wrinklies on the inside. So these are mitochondria. This little top section here is a bud. It's like gonna reproduce, it's gonna break off, become its own guy. Sometimes your animal cells will have like little cilia or flagella, but only sometimes. So here's cilia. They help with movement. What am I forgetting? That everything else is mostly specialized structures. Oh shoot, no, cytoskeleton and stuff. I'm not gonna draw on like a whole cytoskeleton because it would take up the whole thing, but imagine there they are, your cytoskeleton, and that is in plant and animal cells. Your fungal cells have like microtubules. It's microtubules that kind of serve a similar purpose, hence the similar color. Let's see. All right, specialized stuff. In animal cells only, we have centrioles, which help organize the cell for cell division. In plant cells only, we have our chloroplasts. And then in plant cells, oh, well, this is going to look weird now because I left too much space over here. Oh, well, it's okay. We have a large permanent storage vacuole. Why is there a V? Let's think about vacuole. Mainly for water. In your fungal cells, you usually have like smaller food vacuoles. And then last but not least, all the ribosomes. Which we can kind of go crazy with, throw them everywhere. Oh shoot, okay, that one's really ugly. But these are all 80S ribosomes. Because they're bigger than the ones in prokaryotes. Bam, 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 bam. Beautiful. 80S ribosomes. Okay, I don't think I forgot anything, but I also am using like chaos as my guide. So hopefully this was clearer, or at least you can now know what I wrote because I said it to you. Who's to say? But basic animal cell, basic plant cell, basic fungal or yeast cell. Ta-da. Okay. Flippy flip. And so now just to break down like general similarities and differences. Let's make it fun. So here's the plant cell. Dun, 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 dun. Boop. Boop. Animal cells, I always feel like are pink or red because flesh. Dun, 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 dun. And then fungal cell. I don't know what color a fungal cell should be. They'll be brown. They're giving brown vibes. Like a mushroom. Beautiful. Wonderful. Alrighty. To flip backwards in my note sheet. So for plastids, they're like, how to explain this? They're organelles that have outer membranes and then separate sacs. So although mitochondria also have a double membrane, they don't have like internal sacs in the way that like chloroplasts or amyloplasts do. And so plant cells, yes, they have chloroplasts for photosynthesis and amyloplasts for storage of like pigment, things like that. Cell walls, plant cells, yes, they're made of cellulose. Vacuole and vesicle, they have a vacuole, large, permanent, mainly for water. And then they have transport vesicles, centrioles, no. Attachments, no. Animal cells, Plastids? No. Cell wall? No. Vacuole? Very rarely, so we're just going to leave it off. They do have transport vesicles, though. Do they have centrioles? Yes. And then, do they have cilia and flagella? Some do. Example, sperm have flagella. And then fungal cells, last and least. Do they have plastids? No. Do they have a cell wall? Yes. And it is made of chitin. Do they have a vacuole? Yes. It's smaller for food usually. And they also have transport vesicles. They have centrioles? No. But they do have a, and I'm going to butcher this, Spitzenkorper and microtubule organizing centers, which do similar things to your centrioles. They like help it to grow and like reproduce and break off. Then attachments, no. Alrighty. Next, nope, that's backwards. These are just pages that have notes about your organelles. If you don't know the basic function of all of your organelles, please read these. Please. That's like freshman stuff. So like I don't want to spend a ton of time on it, but if you don't have that down, you gotta refresh. Alright. And so, top part of this page is just some basic stuff between prokaryotes and eukaryotes, just like a quick summary of the main differences. I don't know. We'll make prokaryotes purple and eukaryotes blue. Sure. Prokaryotes. Eukaryotes. So for your prokaryotes, let me get the dark purple, so that way it shows up better. Oh, shoot. There we go. So for your prokaryotes, complexity, they are simple, with few structures. For the genetic material, it is in a nucleoid, and it's unorganized. DNA. Size, they are small. They are typically 0.2 to 3 micrometers. Membrane enclosed organelles, no. That's things like, I don't know why I like blanked. Your mitochondria, your ER, your nucleus, your Golgi, your lysosome, your chloroplast, etc. Unicellular or multicellular, they are only unicellular or one cell. Size of ribosomes, small, just like them. They have 70S ribosomes. Compartmentalization, so like that's like your organelles, like your cell can like split itself up and have different areas for different functions. It has like different sub-compartments or like organelles to do different functions. It does not have that. No, it's a very basic, it's a basic little fella. And then examples, bacterial cells. On mine I wrote 69 right here, and I don't know what that's supposed to mean. So we're not going to write 69 right there, because I don't remember what it means. I hope I don't find out. Your eukaryotes, they are complex. They are you. Haha, eukaryotes, it's you. I'm sorry. They have many structures. They have DNA in a nucleus as chromatin. Very organized. Woof. They are large. They're still not big. Like, I mean, it's a cell. They're between 10 and 100 micrometers. Do they have membrane enclosed organelles? Yes, several. Examples: your mitochondria, your ERs, your nucleus, your Golgi, your lysosomes, etc. There's several. Unicellular or multicellular, they could be either. They're uni/multicellular. Their size of ribosomes, they are large. They are your ADS ribosomes. Compartmentalization, yes. And then for examples, they are all plant, animal, and fungal cells. So any human cells, any dog cells, any fish cells, any mushroom cells, any like rhizoid spores, any plant cells, so any pollen, any elodia, anything like that. All of it's eukaryote. And protists. Beautiful. All right, now Scoochy Scooch. This is one that we struggle with. And so these are cells underneath microscopes. Your bottom three down here. Bump ba-da-da. Oof. Bump ba-da-da. This one looks like a tampon. It's not a tampon. It's a bacteria, but it really does look tampon-y, doesn't it? And then these two are eukaryotes. Can you guess which one's a plant and which one's an animal? Take a wild and crazy one. Take your guess. This one is an animal cell. And this one is a plant. And so you're probably like, oh my gosh, Miss White, oh crazy, how did you know? Number one, I'm big brain. Okay. Sorry. Number two, there's also like a couple key structures that like make it obvious to me as somebody who's like studied this for a while. Your cell wall, number one, this like outline around it, I think makes it really clear that this is a plant cell. Like that's my number one look for when trying to determine the difference, is the cell wall. Your other thing would be, at least to me, is the fact that it has chloroplasts. These like, I don't know, big like blobby tubes. These are chloroplasts. The cell wall is supposed to be pointing to the edge. Now I know that it looks weird, I'm sorry. These are chloroplasts. It also has a huge vacuole, which kind of gives it away to me. Your nucleus, your nucleolus. Like, to me this is very clearly a plant cell. And then your animal cell, there's less to see, just because animal cell structures tend to be smaller. But you have your nucleus. There she is. You have your cell membrane. This like white stuff in here is your DNA. It just stains differently. That's like really the only way that you can tell. You could argue that you can kind of see mitochondria separated out here. Kinda. Sorta. Because you can see like the section and then like the ridges in between them. I know it's kind of hard. Your cell membrane you always know is like the outside a little bit. They're saying that you can see ribosomes. I'm not entirely certain. I think that maybe this like little tiny speck might be a ribosome. Maybe. If you like really want to argue, you could also argue that like these like noodly sections here, maybe that's a rough ER. But who's to say? With animal cells, it's a lot less concrete. Pretty much if you don't see a cell wall and you don't see chloroplast, it's probably an animal cell. And then anything you can see after that, great. Just because it's a lot harder. They're just not as clear. Their structures aren't as big, and they don't have as well defined like structures that stain. And so, but what is this? You know? And especially when it's printed on a paper like this. All right, for your prokaryotic cells, you can see your DNA, which is your dark part, or your nucleoid. You can see your cell wall, which is like the light outline here. If you can see a cell wall, you know there's a cell membrane. You can also see cytoplasm. This one you can see pili. They're very light, but they kind of look like little white hairies coming off, kind of. You see what I'm saying? These little pili. You can see your cell wall. And this one, your white parts on the inside are your DNA or nucleoid. And your dark parts are cytoplasm. Should be like whatever stain they used. This one, flagella. Flagella. Okay. And then we have a big boy to look at together. Here's the big boy. Oh, you know what? Now I remember what 69 was. When I looked up, so I was really annoyed, and I was like, what the heck? What are we supposed to label in this one specifically? And so I looked up this image, and it was tabbed on my computer as 69. Because I have this one tabbed too. That way I like make sure I get the accurate structures. Because my computer shows it slightly better quality. Oh no, slightly better quality than is printed on the paper. So here we have our whole nucleus. Dun, 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 dun. Our nucleus. Around it, you can kind of see your nuclear membrane. We have the white DNA. Inside, we have our nucleolus. You probably cannot read that, but I wrote nucleolus, I swear. Right around here, this like very veiny area is one of your ERs. Which one? Who's to say? We'll never know. But it's an endoplasmic reticulum. One of them. Smooth, rough, who knows? But it's one of them. And then this is where me, and when I looked up this image to try to find like the source, we have a couple disagreements, okay? So they're saying that these are all mitochondria because you can see like the ridges. I don't know why I put mitochondria on two lines like that. I don't know. But here's the thing, is that this is a liver cell, and it's specifically a hepatocyte. And so if it's a hepatocyte, those darker circle structures, they should be lysosomes. And so I don't understand why they're saying that those are mitochondria, because they should be lysosomes, but it's fine. It's fine. We're fine. But like these are lysosomes. Like these dark ones, those are lysosomes. And because there's so many of them, that's how you can tell it's a hepatocyte. Like, hello? But whatever. It's my own personal grief with the people who make these packets. I'll forgive them, I guess. They do a lot of work for me, so it's fine. Around this edge, and this edge here, you see your cell membrane. No cell wall. It's an animal cell. And then these dark spots. Oh, where's the orange? We'll make these orange. These little dark spots are your ADS ribosomes. That's about it. That's about all I'd say. I mean, you could label cytoplasm too. Oh, okay. We'll label it 'cause it's going to bother me if it's not labeled. Cytoplasm. Like, that's really about it. For functions and characteristics of life. I need to turn my chair. It's driving me nuts to not, like, write all sideways. There we go. And so, functions and characteristics of life, the most essential things, and the easiest way to remember it, is Mr. Shang. It's not a real person. It's just, like, the acronym. So, M is for metabolism, which is just the idea that living things undertake essential chemical processes are for reproduction. Oh. That living things produce offspring. What is happening? Offspring. Like babies, so sexual or asexual. All living things do that. Hopefully not you until you're older. And hopefully not me, ever. S for sensitivity. Sensitivity. I think I spelled that right. So living things are responsive to internal / external stimuli. So like if I scream in your face, you're gonna flinch. If you are running a fever, you'll feel like chills, you'll feel hot, you'll get like fever sweats. I don't know why that's the thing I came up with, I'm sorry. SH for homeostasis. Living things maintain a stable internal environment. So again, like temperature regulation in humans is a big one. In a lot of aquatic things, like salt concentration in their bodies. Making sure that all your systems are managed. Blood sugar is another really big one. E for excretion. So living things remove waste products. You pee, poo, sweat, snot. Some of us menstruate. It's excretion. And for nutrition, living things, exchange materials and gases with the environment. I was talking about this with the freshmen, like cellular respiration, photosynthesis. You exchange oxygen, CO2. You're eating things. You're excreting them. Okay? You're using all of your macros. We talked about macromolecules and how they're used in the body. Nutrition. And then last up, growth. Oof. Oh, no, I messed up my pattern. Or movement. Living things can move / change shape / size. You can grow. Hopefully you can move to some extent. So can plants. I know it says movement. Plants do move. They just don't like walk around. Okay, but like phototropism, plants do move, and they definitely change shape and size. So, okay, before you have that conversation with me, there's your answer. So these are like the essential things for living. Again, Mr. Shang, I don't know what he looks like. You can look however you want him to look. If it wasn't so late, I would draw you a picture of him, but it's late, and it's already very long. I feel like I skipped a page. I don't think so. I think I am okay. Alrighty. And so then they give you an example. Rude. They give you an example of a unicellular organism. I did skip a page. I turned my page stupid in my book and I lost my page. That's what I get for trying to be fancy. Where the heck is the purge? There we go. And so they give you an example of a unicellular organism that has all of the functions of life. They give you a written out example for a protozoa, and then they ask you to do research about a Chlamydomonas. I'm butchering it. I know I'm butchering it. And so I'm going to ask you all to do this research on your own. Fill in this table as practice. You know what all of these mean. Response is sensitivity. If you get stuck, if you need help, ask me. We can talk about it Tuesday. But for the sake of time, I want to make sure that we cover the things that are slightly harder to like figure out on your own. And so these are atypical cell structures in eukaryotes, or things that kind of go against like our cell theory and like our basic understanding of cells and like eukaryotes. And so there's some of the reasons why there was so much contradiction when learning about cell theory. And so first up is striated or skeletal muscle. And so what happens is you have your muscle fiber cells. They bind together to make these like really long muscle fibers. When they bind together, they form technically one cell that has a single plasma membrane but has many nucleus or nuclei. And so because they fuse together, and they end up having multiple nuclei per cell, and they only have a single plasma membrane. It challenges the idea that all cells, so I guess challenges idea, all cells have one nucleus, and living things are discrete, like separate cell units. The next one they talk about is fungal hyphae. And so they're like these little protrusions. I don't know. Like little protrusions off of fungus. And it's very similar, wherein the cells kind of bond together. And so they have a cell membrane, they have a cell wall. But because they have that kind of like formed together and they're only separated by like septums or like false separations, and their cytoplasm's connected, they end up having multiple nuclei per cell. So imagine like a little like, it's like a dotted line separating them. And so they have multiple nuclei in one cell. And so separated by septums. But they have continuous cytoplasm and many nuclei. What happened there? There we go. So it challenges idea that living things are made of autonomous cells. So they have more than one nucleus, and they have all these cells that are kind of together as one, and they all kind of think and act as one, rather than them each being their own, like, separate thing. If that makes sense. Next up is your red blood cell. And so in mammals, red blood cells don't have a nucleus. They also don't have mitochondria when they get older. And so they end up being produced rather than reproducing. And so no nucleus / mitochondria when mature. They can't reproduce alone. Need to be produced. So they're like a non-traditional eukaryote. And they lack features of, like, I don't know, survival? They know Mr. Shang. They fail Mr. Shang's vibe check. They can't survive on their own. They don't pass. I'm so sorry. And then last up is your phloem sieve cells. And so they have, like, again, tubes. A lot of these are tube-related issues. But they have, like, these cylindrical tubes. And so they're used for transport. They're phloem, so transport of, like, sugars. And so they have these pores, and the nuclei and larger organelles are missing from a lot of the cells in between. And so what happens is that the cells are, like, they need to get things from neighboring cells. Like, they have, like, a little companion cell, a little bestie cell. And so the problem with these sieve tubes is they have no nuclei and few organelles. But they're hollow, which helps for transports. Like, they're really good at what they do. They just need to have, like, a little bestie, like, leach cell to help keep them alive so that the sugar can go through. So here's the sugar moving through. Wow, oh my gosh, so smooth. But they have like a little bestie, like leech cell to help them. Need bestie cell to live. Challenges that multicellular structures have anatomically independent cells. Because they don't. These two are anatomically linked. I feel like I'm getting weird as this video goes on. I'm so sorry. Okay. Next. We're nearing the end. Oh, I like this. Not that I didn't like the other stuff, but like, I like this one specifically. Specifically, this is of high interest to me. Okay. So, endosymbiosis, if you are unfamiliar, is the idea that we once had a bunch of these, like, prokaryotes. Well, I guess if you really want to go all the way back, we can go all the way back to, like, pre-Luca. But we've already talked about that. So we're jumping to, like, post-Luca. And now we have cells, right? We have life. Post-abiogenesis, right? And so we have cells. We have this ancestral prokaryote. At some point, for whatever reason, who's to say, there was this, like, weird infolding of the membrane. Could have been just, like, environmental conditions. Could have been, like, a whoopsie. Who's to say, right? They fold. And then we have this little nuclear envelope forming. Hence why we now have a nuclear membrane. Time goes on. We get more membrane structures. There's my favorite, our little bad boy, our endoplasmic reticulum. Look at him, beautiful membrane structure. And then, here we go. Here's the start of our endosymbiosis. Or I'm sorry, our endocytosis, which is part of endosymbiosis. We have this engulfing or endocytosis of a heterotrophic prokaryote, also known as a purple bacteria. Later to be known as a mitochondria. Crazy. And so we have it get engulfed. It gets endocytosis. I don't know what the past tense of that is. And it, like, kind of becomes one with the team. Because what it does is it does aerobic respiration. So, like, as your cell is able to capture and take in different, like, sugars, foods, like, nutrients, it can do aerobic respiration. So instead of anaerobic respiration, which your poor cell over here has been doing, it's like, oh my gosh, I can do aerobic respiration. 34 to 36 ATP every time. Can't beat it. And so the mitochondrion gets this protection by living within this bigger cell. Now it's like, yes, I'm safe. I get to live in here and get all this comfortableness, and the food just kind of brought right to me. And then your bigger cell gets the energy output from having the mitochondrion there. And so it's like a win-win. So they just kind of chill together. And then that's how we got mitochondria. And then at some point, some of them engulfed photosynthetic prokaryote, or prokaryotic cell, oh my god, aka a cyanobacteria, which can do photosynthesis. And so now, we have the sugars being made on site. We have the ability to do aerobic respiration on site. Now we're cooking with gas. Like, oh my gosh, it's crazy now. Now we have like a full-on plant cell. Now it can fully sustain itself, and it's super effective. It now has a chloroplast, and a mitochondria, and a nucleus, and an endoplasmic reticulum. And this is just like opening up the world for even more and more compartmentalization and development of the eukaryotic cell. And so it's very exciting to me personally, and very fun for me personally. So let's go through, let's highlight our fun new friends. So we have our photosynthetic prokaryote engulfing It's endosymbiosis moment. Look at it go. Get in there. Okay. We have our purple bacteria, our future mitochondria. Endosymbiosis engulfing moment. Oh no, I forgot about her. And her. Oh my gosh. And then we also have our DNA that eventually gets membrane coated by the folding to become our nucleus with our nuclear envelope. It's like the origin story of all of our new organelle friends. It's fun for me. And then we also have our endoplasmic reticulum, who doesn't really play like a huge role in this tale, but because I like the endoplasmic reticulum, we will also include it. The membrane folding, see, here's the membrane folding, look at it. Because as things are advantageous, they're going to continue to persist as whatever the organism is reproduces, right? And so which event, oh, scoochy scooch, which event exactly marks a process of endocytosis? What's the stupid thing? So endocytosis is just ingestion of large particles in a vesicle. So we have one here, and we have one here. And so, engulfing purple bacteria, or I'm sorry, aerobic heterotrophic prokaryote. Just a prokaryote that does cellular respiration. That's all that means. They had to make it wordy. And engulfing cyanobacteria. Or excuse me, photosynthetic prokaryote. A prokaryote that does photosynthesis. Crazy. And so what are the consequences of this? Scoochy scooch. Eukaryotic cells have, get out of here, mitochondria and do cellular respiration. And some have chloroplast and do photosynthesis. And so why is this referred to as endosymbiosis? Endo from endocytosis, the engulfing part of this. And then symbiosis is just like a beneficial relationship between two organisms. And so in symbiosis, both organisms benefit. How does the aerobic photosynthetic prokaryote benefit from living inside the larger prokaryote? Oh, why did I underline that in green? Oh my god. I take it back. How does the aerobic prokaryote benefit from living inside the larger prokaryote? So your aerobic prokaryote, it gets protection, food hunted for it, or like food obtained for it. Either through cyanobacteria or capture. So capture or cyanobacteria. And so it like really benefits. It's like it has like its own little shuttle around places, or its own little personal chef. And then how does the larger prokaryote benefit? I think that's pretty obvious. It gets energy from aerobic cell respiration. What? The purple bacteria. And it gets sugars and food from photosynthesis from the cyanobacteria. It's a win-win. They're hyped about it. I'm hyped about it. They should be too. Alrighty. So to summarize our steps that we took, step 1, we have, folding membrane, which results in nucleus and ER. Step 2, we have endocytosis of, oh geez Louise, purple bacteria, or our aerobic heterotrophic bacteria. Step 2A, optional, endocytosis of cyanobacteria or our photosynthetic prokaryote. And then step 3 to getting a mitochondria and a chloroplast is evolution and profit. They evolved over time to get the slightly more adapted structures that they have now, and because they all benefited and they all profited from the situation, it kept happening. Ta-da! And so we have purple bacteria. There she is. I don't know where the mitochondria is supposed to be in that picture. It's too small. We'll just pretend like it's, I think it's this, maybe. It's hard to tell. I think it is that. I think. It's really hard to tell. I really genuinely don't know. And then cyanobacteria. Meow. Meow. This one, I have no clue. I'm just gonna guess it's this. Sure. Oh, I guess I should... Chloroplast. Keep it nice and consistent. Oh. Mitochondria. Alright, so then down here it gives you some information about all of them. And so what things do they have in common to help prove that this whole endosymbiotic theory happened? And so eukaryotes, they have DNA organized into chromosomes, but prokaryotes have a single loop, which is the same as mitochondria and chloroplast. I need 2 colors. What's the color I haven't used? We use blue. And then for ribosomes, eukaryotes have 80S, but look, prokaryotes 70S, mitochondria and chloroplast 70S. Size in microns. Oh my god. Large, small. Has a nucleus? Yes. Has a nucleus? No. Membrane-bound organelles? Yes. Membrane-bound organelles? No. Because these are some of the membrane-bound organelles, so like, duh, they don't have them. Number of replication, mitosis. These ones all do binary fission. And then replication of number of cells, 1 or more, usually 1, and then like the irrelevant, nope, not applicable, NA. I dropped this lid. I'm losing my mind. Where's this lid? Oh, it's in my shoe. I took my shoes off. It's information you really didn't need to know. Alrighty. So summarize our findings. There's like 5 key bullet points. And there's one that's like not on here. That bothers me. So key findings. Why are they similar? Oh, please. Number 1. Mitochondria and chloroplasts have own genes, and they have circular DNA like prokaryotes. Two, have 70S ribosomes like prokaryotes. Three, reproduce by binary fission like prokaryotes. 4, can only be produced by division of pre-existing mitochondria / chloroplasts, meaning that they're like unique DNA. And then last up, and probably like the most important, where's the teal? There we go. 5, they have a double membrane. So they have like the OG membrane from living as a bacteria. Plus additional vesicle membrane from endocytosis. It's like the easiest evidence for it. How else would they do that? All right. on to the next ones. Oh, wrong way. And so for these practice questions, I would like you to try these on your own as like a proof of you understanding cell theory. For cell differentiation to develop specialized tissues, we will talk more about specialized cells in class, so I'm not going to spend too much time talking about specialized cells. It's pretty like clear, it's just cells will develop slightly different structures. And so like sperm cells, they have flagella to swim to the egg. Muscle cells, they have that whole striated business going on. Other easy ones, blood cells produce more hemoglobin. Nervous cells have protrusions, send signals. It's basically when their structures adapt to help better suit their function. Okay. And so as organisms learned that adapting their structures helped, they also learned that adapting together to be able to better have more and more of these specialized structures and have them be able to live together was really beneficial. And so we developed multicellularity from unicellularity. And although a lot of organisms are still unicellular, there are benefits of multicellularity, one of which being a longer lifespan. One cell dying does not mean death of organism. Their organisms can be larger. They're not limited by surface area to volume ratios. We'll talk more about that with cell membrane. And then you can have higher complexity in your organism, not only specialized cells, but special cells working with other special cells. And so you end up having like the ability to have cells that only focus on one thing because you have a whole another set of cells forming a tissue that focus on a fully different body function. And so you're able to have much more complex organisms. Think about your complexity versus the complexity of an amoeba. And then there's examples down here, and then I'll also link a video in the description. But yeah, that's it. You have DBQ questions on the back to do. And that's it. Thanks. This is obnoxiously long. I'm so sorry. It's 7:30. I want to go home.